The overall goal of this procedure is to demonstrate a method for the production of metastatic tumors in black 6 mice.
This method can help answer key questions in the field of immunotherapy, like how investigative agents affect the immune response in the lungs. The main advantage of this technique is that it's experimental metastases, so the results are reliable and relatively consistent.
To begin, place cultures of B16-BL6 murine melanoma cells, which should be at approximately 70% confluency, in a sterile hood. Next, add 1 milliliter of 0.05% trypsin EDTA to each dish and leave the cells for 1 minute. After aspirating this solution, proceed to add 1 milliliter of trypsin to every plate and then return the cells to the incubator. After 10 minutes, move the cells to a sterile hood and add 4 milliliters of serum-free rPMI medium to each plate. Then, collect the cells with a sterile motorized pipette. To break up clumps that could potentially clog the ejection needle, press the tip of the pipette against the bottom of the plate and expel the cells. After repeating this process several times, recollect the cells and transfer them to a 50 milliliter conical tube. Then, remove a sample of the cell suspension, introduce it into a hemocytometer, and determine the cell density. Equally aliquot the cell suspension into 4 15 milliliter tubes.